Hi students, welcome to our creation. So this is Rajesh, MSc Zoology. Today we are going to discuss about important topic of genetics, multiple alleles. Before going to know about multiple alleles, we have got some idea on the some words of genetics like gene. What is gene? Gene is a sequence of DNA which control the phenotypic trait like skin color, eyeball color, hair color, whatever it is. Gene is a sequence of DNA which control the phenotypic trait. And the another word is allele. Alleles are the alternative forms of gene. For example, there is a trait length of plant. Length of plant. If it is a trait, it have two alternative forms, either tall or short. Likewise, a gene has two alternative forms like called alleles. For example, it is the control of gene. T is indicating the tall plant and small t indicating the small plant, short plant. This is the dominant over this character. These two are called alleles. Alleles are the alternative forms or alternative versions of a gene. Gene is a sequence which control the phenotypic trait. Now, come to the another word, dominant character. Dominant. If the two alleles are like this, both are indicating tall plant, so tall plant should be produced. If the alleles are like this, both are indicating short plant, so short plant will be produced. Now, what is the fate of this genotype? What is the fate of this genotype? In this genotype, capital T, this is the identification symbol of tall plant. This is dominant character. This is dominant over this character. So tall plant should be produced. Here, capital T is tall is dominant over small t, dominant over short plant. This is the dominant character and this is the recessive character. Here, if the both characters are equal, run to capital T, both are capital T, so it is known as homozygous dominant. Here, both are recessive, it is known as heterozygous recessive. Homozygous dominant, homozygous recessive, homozygous recessive, homozygous dominant. This is known as heterozygous because both are unequal. Heterozygous dominant. Now, these are the basic concepts to understand multiple allelism clearly. These are the basic concepts to understand genetics clearly. Now, come to the multiple alleles. You already know about this. Each and every gene have two alternative forms called alleles. But some genes may have more than two allelic forms known as multiple alleles. Some genes may have more than two allelic forms at the same locus of the homologous chromosomes. Homologous chromosomes like this. This is the locus of allele. The alternative forms alleles. This is from mother and it is from father. So a gene have two allelic forms at the same locus of homologous chromosomes. At sometimes some genes may have more than two allelic forms called multiple alleles which are present at the same locus of homologous chromosomes of a population, not an individual. So multiple allelism, the phenomenon multiple allelism we can observe only in population, not in individual, not in single individual. We can observe this multiple allelism only in population. Now, come to the topic multiple allelism. We can calculate the genotypes of the alleles by the equation n into n plus 1 by 2. N into n plus 1 by 2. If the alleles are 2, if there is 2 alleles, the 2 alleles can produce how many genotypes? in this equation, n is number of alleles. So 2 into 2 plus 1 by 2. Both are cancelled. 2 plus 1, 3. So if two alleles present of a gene, two alleles can produce three types of genotypes. For example, P and T. These are the two alleles. They can produce three types of genotypes: homozygous dominant, heterozygous dominant, and homozygous recessive. These are the genotypes. So we can calculate the number of genotypes on the basis of number of alleles. If the person give number of alleles and count and ask count the genotypes, we can count the genotypes by this equation. Okay. Now come to the multiple allelism. The well-known example for multiple allelism is ABO blood system of human being. Before going to know about the generic basis of ABO blood system, we must have the general idea of ABO blood system. We may learn about ABO blood system in our previous classes, like ABO blood system is proposed by Karl Landsteiner. ABO blood system was proposed by Karl Landsteiner, who is Nobel laureate in 1930 for his work on medicine. Right? He proposed a theory of ABO blood system, a blood grouping, on the basis of presence or absence of antigens on the surface of RBC. Here, this is RBC, and it is a plasma membrane of RBC, which is lipid layer. Here, some carbohydrate polymers or polymer sugar chains present on the plasma membrane of RBC. These are the antigens or sugar polymers which are present on the surface of RBC. On the presence of these antigens are of two types. One is antigen A and another one is like this. It is B type. There are two types of antigens present on the RBC surface. Based on the presence or absence of these antigens, he divided the blood groups into four types. Likewise, if A type of antigens present on the surface of RBC, the blood group is known as A group. If the B type of antigens present on the RBC, the blood group is known as B group. If the person have both A type of antigen and B type of antigen, he belongs to AB blood group. If the person doesn't have any of these two antigens, either A or B, any of these two antigens are absent on the surface of the RBC, he belongs to blood group O. This is the theory of Karl Landsteiner. According to this theory, there are some natural antibodies present in plasma. A group of person have anti-B antibodies. Anti-B antibodies are against to antigen B. A group person have anti-B antibodies in their plasma. B group persons have anti-A, reverse, anti-A antibodies in his plasma. Well, come to the AB blood group. In the AB blood group, the plasma doesn't have any of the two antibodies, anti-A and anti-B, both are absent. So there are no antibodies present in plasma of AB blood group person. There are no antibodies present in AB blood group person. So there are no fighters. So he can receive any blood. So he is called universal recipient. He can receive any of these 4 blood groups. And come to the O group, there are no antigens fight against another group in O group. And the plasma of O group have both anti-A and anti-B. Both anti-A and anti-B antibodies are present in a O group person. These are the 4 types of blood groups and 2 types of antigens, 2 types of antibodies. According to this theory, there are 4 blood groups are present on the basis of presence or absence of antigens. Antigens are the sugar polymers which are present on the surface of or which are embedded in the lipid layer of RBC. The antigens are also known as isoagglutinogens because they cause agglutination, and antibodies are also known as isoagglutinogens. This is the general idea about ABO blood system. Now we are going to know about the genetic basis of the ABO system. Bernstein is the scientist who discovered the genetic basis of ABO system. According to his, there is a gene which controls the ABO blood system in human beings is I. The gene name is I, which is present on the 9th chromosome. According to his, he discovered that a gene controls the ABO blood system of human beings. The gene name is I. I is present on the 9th chromosome. The gene I have 3 alleles, 3 alternative forms. They are IA, IB and IO. The gene I have 3 alternative forms. So it is multiple allelism. It shows the multiple allelism. So it is the best example for multiple alleles. Bernstein discovered that a gene called I is controlled ABO blood system of human beings, which is present on the 9th chromosome. The gene I have 3 alternative forms called alleles, IA, IB and IO. IA can produce antigen A on the surface of RBC. IB can produce antigen B on surface of RBC. IO cannot produce any of these 2 antigens. And here IA and IB both are dominant over IO. And I and IB are codominants. This is the equation of IA is equal to IB greater than IO. IA and IB both are codominants, and these 2 are dominant over IO, and these 2 are dominant characters. This is residue character. Now come to the genotypes of these antigens. Now, how many genotypes can be produced by these 3 antigens? There is an equation, n into n plus 1 by 2. There is an equation, 3 antigens. In this equation, n is number of alleles. Three into 3 plus 1 by 2. 6 genotypes can be produced by these 3 antigens. The 6 are IA, IO, IB, IO, IB, and IO. Here it is homozygous dominant A. It is heterozygous A. Because A is dominant over IO. It can produce antigen A. It also can produce antigen A. Because IO is residue character. This is dominant character. So antigen A is produced by this genotype. Now, here antigen B is produced. Here also antigen B was produced. These are the antigens produced by these genotypes. In this condition, both A and B are codominants. So antigen A and B both are produced. Here both are recessive. So neither A nor B. Neither A nor B. So no antigens are produced. This is the genetic basis of ABO blood system. There is a gene called I which have three alleles IA, IB and IO. Here IA and IB are dominant over IO and IA, IB are codominants. These three alleles can produce 6 type of genotypes. IA, IIO both are produced A type of antigen, and IB, IBO both are produced B type antigen. IB both are codominant, so A and B both antigens are produced. Here this is recessive character, so and no antigens are produced. Let's draw a table of blood group system. Genotypes. How many genotypes are present in this theory? IA and IA, 6 types of genotypes. Write all the 6 types of genotypes. These are the genotypes. It can produce antigen. Which type of antigen can be produced by this genotype? Antigen A, antigen A, antigen B, antigen B. These two are codominant, so antigen A and antigen B, not AB, A and B. Here, no antigens are produced. It is not O, it is no. Now, if A type of antigen present on the surface of RBC, the group is A group. It is also A group. This is B group. It is also B group. This is AB group, not A and B group. Here A and B antigens. This is group, AB group. No antigens are present, so it is called O group. Don't write O here, O group. These are the antigens, these are the groups. And now, antibodies. If a person have A type of antigen on the surface of RBC, which type of antibody is present in his plasma? In his plasma, anti-B type of antibodies are present. Just reverse. Anti-B type of antibodies are present in antigen A blood group. Here also anti-B antibodies are present in plasma. Now, anti-A type of antibodies are present. Here also anti-A. If the person have both antigens A and B in his blood group, which type of antibodies are present in plasma? There are no antibodies. There are no antibodies in his plasma. Now come to the O group. Here there is no antigens. There is no antigens. So in his plasma both antigen anti-A and anti-B, both the antibodies are present. Anti-A and anti-B. Anti-A and anti-B. Now which groups can tolerate by these groups persons? He can tolerate A group and O group. First of all, we know that O is universal donor. O is universal donor. So he can receive, all the persons can receive blood from O group person. So write first O. All the persons can receive blood group from O. Now A person can receive blood from only A and O. he also A and O. The person who have B group can receive blood from B and O. The person who have AB group in his blood can receive all the blood types because there are no antibodies. There are no antibodies in his plasma to fight. So AB and O. Now O person can receive only blood from O only. He receives blood from O only. In the transfusion, he can donate blood to. He can donate blood to A person who have blood group A and AB, because AB, in the AB blood group there are no antibodies to fight against A. So AB is universal recipient. Write the AB in all the groups. AB can receive blood from all the groups. Now A can transfuse blood to A group and AB group. B can donate blood to B and AB. AB can donate blood to AB only. AB only. Because if he donate blood to A group, there are anti-B antibodies to fight against. If he donate blood to B group, there are anti-A antibodies to fight with AB group. If he donate blood to O group, there are anti-A and anti-B. Both antibodies are fight against AB group. So he can donate only to the AB group only. Now O can donate A group because there is no antigens. B group, no antigens. AB group and O group. So he is universal donor and he is universal recipient. This is the multiple allelism which belongs to ABO blood system. Thank you.